This is the plasma membranes chapter. So the first thing to take note is that there is a difference between passive transport and active transport, where passive transport uses no energy required, to allow molecules across the plasma membrane, where active transport actually requires energy to transport molecules across the semi-permeable, across the plasma membrane. Let's start with the passive transport section. We'll talk about diffusion, we'll talk about osmosis, and we'll talk about facilitated diffusion. Say we put in, say we have a cup full of water. We have in here a semi-permeable membrane, which means that some things can get through, some things cannot. And in this case, well, let's say that water can get through, and where if we put salt, drop salt in on the left-hand side of the semi-permeable membrane, the salt cannot get through, but water can get through. If water can get through, and we're talking about water, then this is the phenomena osmosis. Osmosis is only the diffusion of water across a membrane. So the first thing you ask, okay, you put in a whole bunch of salt. The question is, which way is the salt going to move? Which way is the water going to move? Well, the salt will not move because it can't get through the semi-permeable membrane. The salt is too large of a molecule, so it can't get through. But here's how you can ask, here's the question you always want to ask, is which way does the salt want to move? Well, the salt wants to move, and the fancy term is down the concentration gradient. Because these guys, just like if you take a bottle of perfume or cologne and you spray it, the smell is very, very concentrated right around the bottle, and then the molecules will want to diffuse to space themselves out equally. That's what diffusion is, the tendency for molecules to spread out evenly into the available space. Well, it's the same idea. These salt molecules or crystals want to diffuse, and so they want to move down the concentration gradient, which means from high concentration to low concentration. But they can't. They can't move. Water does move, and so water moves in the opposite direction. If salt wants to go from high concentration to low and can't, water will then move the other way. Why? And so the question is why? Why will water move from the right to the left? If water moves from right to left, it will allow these molecules to be more spaced out because you're putting in more water, and so that these molecules will then be more spaced out like the ones on the right-hand side. What happens to the level of the water then? Well, if water is moving from the right-hand side to the left-hand side, the level of the water on this side of the membrane will then move up. And as you can imagine, as this continues, these salt crystals will continue to space out more and more because now there's more and more water, and there you have it. That is osmosis. The first question you ask is, which way do the salt molecules want to move? And the answer for that would be from the left to the right, and therefore water moves in the opposite direction to try to space out those molecules. The salt wants to move from high concentration to low concentration, and that's called moving down the concentration gradient. This is an example of diffusion, and more specifically osmosis, where we're talking about the movement of water across a membrane. And this again requires no energy because we're going from because the water does not require energy to move across the membrane. Okay, now in this example here, let's say this is a blood vessel, and here is one here's one red blood cell. Here's one red blood cell. Relatively speaking, it looks like there's more solute inside the red blood cell than outside the red blood cell in the stream. The red blood cell in this scenario is said to be hypertonic. The word hypertonic refers to the solute, and relatively speaking, it is hypertonic to the outside of the cell, or the extracellular. So in this example, this red blood cell is hypertonic. You can also say that that extracellular fluid is hypotonic to the red blood cell. Why? Because there is less solute outside than there is inside. There is more water outside than there is inside, but this tonicity is not referring to the amount of water, it's referring to the amount of solute. What is solute? What is solvent? And what is solution? Solute would be like the salt here, the solid substances. Sugar, salt, those would be examples of solute. Solvent would be the liquid that is also in the same area. This example would be water. Water we generally use as the solvent. Water would be solvent, salt would be the solute. Altogether we would call a solution. This altogether we would call a solution. So the outside of the cell is hypotonic to the cell, and the inside of the red blood cell is hypertonic to the outside. And if this occurred, the question would be, how would this even come to be? Well, you drink something that has a lot less salt than what you would normally have inside the red, or some sort of solute inside the red blood cell. If you drink a whole bunch of pure water, for example, or distilled water that has absolutely no solute inside, you would have this sort of scenario where you would have a lot less stuff. And so now the next question is, which way? It would be the same sort of question. Which way does the solute want to move? Well, you have high concentration inside and low concentration outside, so the solute wants to move out. But it can't, because this is like a semi-permeable membrane. This is the plasma membrane of the cell. The solute wants to move out, but it can't. Therefore, the water moves in. And since the water moves in, the last question you would ask is, what happens to the cell? Does the cell swell up, or does it shrink? Well, if water is moving in, this thing will continue to swell up and might eventually lyse and break apart. So now we've talked about hypertonicity and hypotonicity. If the solute outside is the same as the solute inside, if we took away some of these, and the distance between each of these particles would be more or less the same, like that, then you would say the cell is isotonic to the extracellular space, or outside is isotonic to the inside. Isotonic, hypertonic, and hypotonic. This is all passive transport. This requires no energy, and this is again osmosis, where we are talking about water being able to flow through a semi-permeable membrane at any point with no energy. In facilitated diffusion, it's almost the same idea, except there will be little proteins where molecules have to go through those proteins. It's also passive, where it requires no energy, but instead of molecules being able to travel through the membrane at any point, at anywhere throughout the membrane, the molecules can still get through. However, they require to get through particular points of the membrane where you would have specific proteins, and then they can get through still freely, but they need a particular protein, so it's called facilitated, and that's here, facilitated diffusion. It's still passive, requiring no energy. Okay. The next thing we're going to talk about is active transport. Active transport does use energy. There is even more examples than just these. However, we'll talk about the sodium-potassium pump, co-transport, endocytosis, and exocytosis. So, here is an example. Here is an example of a semi-permeable membrane. It is semi-permeable. This is a phosphobilipid layer. Each of these molecules is one phospholipid. You have the hydrophilic head and two hydrophobic tails. And these phospholipids just orient themselves because the hydrophilic end likes the hydrophilic water on the outside of the cell and on the inside of the cell. And the hydrophobic tails fear the water, and so that's why these phospholipids orient themselves the way they do. These are how all plasma membranes look like. So we call it phospholipid bilayer because there are actually two layers here. All of the inside is hydrophobic, and the small hydrophilic heads are on the outsides. Now, in active transport, and the specific example, the sodium-potassium pump, this requires energy. Why would anything require energy? And the answer is because we are trying to go from low concentration gradient to high concentration gradient. These molecules want to move from high to low. They want to diffuse and space out it equally in the available space. But if a particular protein or a cell needs to transport molecules against concentration gradient, or from low concentration to high concentration, that's the opposite way that the molecules want to move. And therefore, those particular proteins require energy to pump them against the concentration gradient, getting them out of the cell or in the cell, whatever happens to be the case. There is also co-transport. In co-transport, you have a couple of proteins involved. You have a proton pump, and what do you notice? The proton pump is pumping hydrogens, would you say, from high concentration to low concentration, or from low to high? The answer is from low to high. This is against concentration gradient. Well, and that requires energy. That requires energy. So, why would any protein want to do this? Now you have yourself a high concentration gradient And these protons are going to want to diffuse back. And in doing so, they go through a different protein, and along with them, they bring along with them a molecule that can't normally get through. For example, glucose, sugar. And that's one way you can get sugar into a cell. How do you do that? Sugar is a pretty large molecule. How do you do that? You use co-transport, because those protons can now bind with the particular molecule that's trying to get in, and allows the movement of those larger molecules that wouldn't normally get in. All you have to do is take some energy, you make a high concentration gradient, and therefore those protons are going to want to get back in, and they bring along with them some molecule like glucose or sucrose or fructose that's larger than what is allowed through, and that is called co-transport. Co meaning combined. Combined. Endocytosis is the word cytosis comes from the word cell. Endo means to take in, so anything that comes into the cell is technically endocytosis. Endocytosis. And this is generally, we're talking generally about things coming in bulk. Endocytosis. And there are three types: phagocytosis, and pinocytosis, and receptor-mediated endocytosis. Phagocytosis is eating, engulfing, eating large molecules. Pinocytosis is drinking, cellular drinking, and receptor-mediated endocytosis is taking in molecules but also require some sort of receptor to allow the molecule to get in. If this were a cell, and we have some sort of molecule that needs to get into the cell, as this gets closer, the cellular membrane now begins to engulf, like these little pseudopods, and begin to engulf the molecule, just like that. The plasma membrane is extremely fluid. So in just a short while, the cellular membrane, plasma membrane, will be able to engulf, and now you have inside, in just a short while, now you got yourself a little vesicle with what you engulfed, and that would be an example of phagocytosis. Pinocytosis works the same way, except instead of being some large molecule, it's drinking of some kind of solution. And molecules dissolved in droplets are taken up when extracellular fluid is gulped into tiny vesicles. And finally, receptor-mediated endocytosis, you would have the same sort of thing, except now you have these little receptors all along the plasma membrane, and you have these fitting little molecules. When the receptors bind to these molecules, these little ligands, the cell begins to engulf and take in. They don't know how to do that without these receptors. When they bind, when these little molecules bind, then that allows the cell to know, okay, let me take this in, and so the cell begins to engulf these, and eventually it'll look like this, and eventually it'll look like that. It's the same idea as phagocytosis, except the molecules themselves need to bind to the receptors on the plasma membrane, and that's why it's called receptor-mediated endocytosis. So that is sodium-potassium pump, co-transport, endocytosis, and the three types of endocytosis are phagocytosis, which is cellular eating, pinocytosis, which is cellular drinking, and receptor-mediated endocytosis, which is the taking in of molecules but using receptors on the outside of the plasma membrane. And the last thing is exocytosis. Exocytosis is the opposite, exiting, things exiting the cell. And it really works the same way. You have a package that needs to get out of the cell. The package is wrapped around in a vesicle, and the vesicle, the little package itself, the envelope, is made out of phospholipids. And because it's made out of the same stuff as the plasma membrane, once it reaches the outside, the plasma membrane, it's able to fuse and join, the phospholipids can join the other phospholipids, and so the envelope essentially joins with the rest, all those phospholipids in the package joins the rest, and eventually that package becomes, the envelope itself becomes part of the plasma membrane on the outside of the cell, and therefore now the contents inside of that envelope is released to the outside of the cell. That is exocytosis. It's really just the opposite of endocytosis. Instead of taking in, you are taking out. So that is the summary of plasma membranes.